Okay, so welcome everybody. Thank you for joining this session. So I think you hear me all right, right?
Yes, yes, your
Yeah,
audio is so
yeah,
good.
yeah. All right, thanks. So I'm Ramhari. I'm a postdoc at the Mendel Lab in the Department of Biological Sciences. So today I'm very happy to share my research work in Centrapalus pauciflorus, so which is a potential novel industrial oilseed crop. And then I'll be talking about the phylogenomics and population genetic study of the Centrapalus. Um, all right, so Centrovirus pauciflorus are tropical annual herbs, so they have indeterminate type of growth. So that means these plants keep on growing and producing new flowers and fruits throughout their lifetime. And then these existence or Centrovirus, they are usually found in the Eastern African countries, so from Ethiopia to Mozambique, and they are not cultivated. So that means they grow as a weed in the valleys, usually in the disturbed field along the roadside and riverbanks or in the forest. All right, so why do we care about this plant? Because the seeds of Centrapalus, they are rich in naturally epoxidized oil. So with 70 to 80% of the oil contained being vernolic acid. So vernolic acid is a long chain fatty acids containing an epoxide ring, as you can see here. And then vernolic acid as a plasticizer and stabilizer finds many application in industries such as plastic, rubber, paint, cosmetic, and pharmaceutical industries. So in this picture, these seed-looking structures, they are in fact the fruits of Centrovirus versiflorus, and the common name for such fruits is the achene. All right, so plasticizers are extensively used to make plastics or rubber flexible. They are also used as solvents in cosmetics, as coatings in pharmaceutical products and others. So globally, 7.5 million tons of plasticizers are consumed every year, with 80 to 90% of that being used in plastic-related industries. In Europe alone, almost 1.5 million tons of plasticizers are used every year, especially in the plastic-related industries, as shown in this chart. So we can see that like 20% is consumed to manufacture wires and cables, 20% to flooring and wall covering materials, etc. All right, so there are so many chemicals used as plasticizers, but if you take a look at this graph, um, so different forms of phthalates, so which are synthesized by modifying petroleum products, they have been extensively used as plasticizers over the years, because that is also because they are highly compatible with the plastic products. So, but the bad thing is these phthalates are associated with different health hazards in both children and the adults, and much research has been done in this topic. So, but the good thing here is the use of, even though at a very small scale, the use of plasticizers over the years has been reducing and replaced by some other form of plasticizers. All right, so as an alternative to phthalates, plasticizers manufactured by artificial epoxidation of animal fats or vegetable oils are also gaining grounds. But naturally epoxidized oils such as vernolic acid are far superior than them in the sense that they are less viscous, they are non-volatile, and then environment friendly. So, uh, uh, but commercial production of epoxidized oil has not been achieved yet, even though the potential sources such as Centrovirus, pauciflorus, and Euphorbia legasii were known way back. Okay, so attempts to grow Centrovirus pauciflorus commercially in Ethiopia and North America have not been successful because the seeds do not germinate well, and then they do have tall plant height, as you can see in this picture, and then the seeds also mature randomly, so this picture shows that only the 50% of the seeds are mature, where some of them are still in the flowering stage. And there is also seed scattering, so which means that so whenever the seeds mature, so they fall to the ground and the harvesting becomes difficult. Uh, but due to its economic importance, there was a planned exploration effort from USA throughout Africa in 1980s, which collected 37 excisions of Centrapalus pauciflorus, and those collected seeds are stored in the two labs in Arizona and Iowa. So some of the original seeds are grown, multiplied, and stored, and also made available to the researchers for research purpose. And then, so also the information regarding the seed collection date, places, collectors, etc., are available in the USDA website. So with all those resources available for an important potential industrial oilseed crop, the general objective or the central question for our research is to study the evolutionary relationship among different Centrovirus excisions and assess their genetic variability and population structure, so that the outcome of the study will be helpful towards establishing Centrovirus as an industrial oilseed crop. Okay, so more specifically, first we'd like to study the relationship among those available Centrovirus excisions using their DNA sequences by producing phylogenetic tree. If you are not familiar with phylogenetic tree, so this can be produced by using morphological as well as molecular data such as DNA sequences or protein sequences. So as you can see that they have different parts like branch, roots, node, and these parts, they have their specific message, but as a whole, the tree is very informative for evolutionary studies, evolutionary and other studies. Basically, they tell us the relationship among the study excisions. For example, here this gorilla is closely related to rat compared to lizard because it shares the common ancestor with rat at this point of time, so much more recently than with the lizard way back here. So we can use this type of information in our research areas too. For example, if this tree is a Centrovirus tree, and then so this Centrovirus excision 1 has a very important agronomic character, let's say high percentage of vernolic acid in the seeds. So it is highly probable that this Centrovirus excision 2 also has the similar characteristic, right? Or let's say this Centrovirus excision 4 is a disease-resistant variety, and then at the same time, it is highly possible that this is Centrovirus excision 5 also has that character, or maybe so that character is present in the whole clade, including Centrovirus excision 3, 4, and 5. Okay, so this phylogenetic trees, they help us for the selection of specific, let's say, specific excision of Centrovirus that could be developed into a successful crop. All right, so phylogenetic trees, there are so many other applications of phylogenetic trees, and evolutionary biologists are always happy to talk about those. All right, so next we would like to develop a novel genomic tool or microsatellite markers specific to Centrovirus species, so or Centrovirus excisions. We know that sequencing is getting cheaper, but at the population level, studying at the population level where there will be hundreds of individuals, so sequencing is still expensive. So in those cases, like these microsatellites come very handy. So actually, these microsatellite markers are the short sequence repeat regions in the DNA of the organism. So here we can see that there are 5 repeats of 4 DNA bases like TAA, and then so we identify these microsatellite regions in the DNA using some software programs, and then develop the primer so that we can amplify them, and then compare them among different individuals for population genetic studies. And because these sites are often variable among individuals of the population or also among different populations. All right, so finally, we would like to assess genetic diversity and population structure among Centrovirus excisions. This helps us to identify the Centrovirus excisions which have high genetic variability, and those variabilities might be useful for converting into the crops. So we can also do PCOA or structure analysis. So for finding out how many distinct genetic clusters are present in our study populations. So as an example, in these two plots, So the PCOA as well as structure, we can see three genetically distinct clusters. Here, the small dots or the characters, so they represent individual in a population, whereas the bars over here also represent the individual in population. And these genetically distinct clusters, they are color coded. All right, so our study model, Centrapalus, belongs to the plant family Asteraceae. So family is a group in the hierarchical system of classification. And then, so this Asteraceae family, which is also called the sunflower family, is considered very successful, and the members belonging to this group grow in all sorts of habitat. So in fact, 1 out of 10 flowering plants belong to this group. And one of the most important characteristics of this group is that the flowers are arranged in a head or structure. Flowers are arranged in a structure called head or capitulum. So that means these single flower looking structures in the members of Asteraceae, in fact, they are the collection of many flowers. All right, and then this family Asteraceae is divided into further smaller groups, which are color coded here. And this diagram, this tree, phylogenetic tree, is taken from the work by Mandela and group in 2019. All right, so Gilbert in 1986 divided the Centrovirus accessions, then known as Vernonia gallamensis, into two broad groups based on the flower structure and the associated parts. And he further divided this Vernonia gallamensis or Centrovirus pauciflorus group further into 6 smaller groups called subspecies, and the subspecies gallamensis was further divided into 4 other smaller groups called variety. So to summarize this relationship, so we have all the Centrovirus accessions, then known as Vernonia gallamensis, divided into 2 broad groups, with this group further divided into 6 subspecies, and further this subspecies divided into 4 varieties. All right, so 2 years later, so Jeffries slightly modified the relationship. For example, he put all the Centrovirus accessions into 1 major group instead of 2, and then divided that further into 6 subspecies, and further dividing 2 of the subspecies into varieties. So for those of you not familiar with taxonomy, so this might look very complicated, but just trying to make this easy. For example, if we have this Centrapalus accession, it could be either like Centrovirus pauciflorus subspecies Phyllisquama, or it could be like Centrovirus pauciflorus subspecies Metamoensis, or based on its character, it could be like Centrovirus pauciflorus subspecies Pauciflorus variety Peditiana. All right, so again, in 1999, so Robinson found other plant accessions which look very similar to Vernonia gallamensis, and then he grouped all those similar looking plants into a separate group called Centrovirus. Since then, Vernonia gallamensis has been identified as Centrovirus pauciflorus. But later in 2010, all those similar Centrovirus looking accessions are further based on their geographic distribution and further morphological characters, they are transferred into the new group called Vernonella. Okay, so this is the latest classification system, classification for our study model Centrapalus. But again, this classification is not well recognized by other institutions such as the Plant List or IPNI. So that means we have confusions regarding the nomenclature, regarding the classification, and regarding the relationship among different accessions of Centrapalus. So which is, we know, a very important potential novel industrial oilseed crop. So so that's why it also gave us the opportunity to study the Centrapalus as a whole, and we came up with as stated earlier, we came up with the following objectives. Like, we would first like to draw the phylogenetic tree to show the relationship among different Centrapalus accessions. Then we would like to develop a genomic tool so that we can study the populations of Centrapalus accessions, and then finally perform the genetic study, the genetic diversity and population structure of some of the populations of Centrapalus accessions. All right. So for the methodology, we needed for producing the tree, we needed the Centrapalus accessions. For that, we collected seeds from the USDA, and then we grew them, many of them, in the greenhouse of the University of Memphis. And for some other Centrapalus accessions, we collected the leaf tissue from the Smithsonian Museum for the DNA extraction. All right. So for the methodology, we use the high-seq technique, so which is a next-gen sequencing approach combined with bioinformatics. So for the protocol, we followed this paper by Mendel and group in 2014. Uh, so in short, so we targeted, captured, and sequenced 1,061 genes from each Centrapalus accessions and compared those DNA sequences or DNA from them to produce the relationship among those Centrapalus accessions. So in form of phylogenetic tree, which were built by using programs such as RexML and Astral. So basically here we are comparing the DNA sequences from Centrapalus accessions and then trying to find the relationship among them. All right, so just to give you an idea, like so this picture is the comparison of the DNA sequences from Centrapalus accessions and then a few other closely related plant accessions to Centrapalus. And then even though we targeted 1,061 genes to compare among these study accessions, we after some bioinformatics pipeline, so some 700 genes were available for further analysis. And so the combined length of all the genes from 1 to 700 was more than 500,000 DNA bases. So this is just a zoom in portion of this data matrix. And then it's so nice and interesting to see that how similar, how closely related accessions share the same DNA, base in the specific region in the DNA sequences. All right, so this is the tree which is produced by RexML analysis, which uses maximum likelihood algorithm. And then here, what we see is all the Centrapalus accessions, they cluster together in a group, which tells that based on their DNA sequences, they are closely related to one another, and they are different not only from the other distantly related accessions belonging to different classification groups, but they are also different from some of the accessions belonging to the same classification group, Centropalini. So within the Centrapalus group, so there were two distinct classes, one belonging to the subspecies pauciflorus, and then the other belonging to the three subspecies of the Centrapalus. So we didn't have a sample from these accessions then. All right, so again, so it was interesting to see that the Centrapalus looking accessions, as stated earlier, those were transferred into the new group Vernonella. So based on the geography and then their morphological character. So that was also supported by our DNA data in our study because they came to lie outside. They formed their own group outside the Centrapalus group and didn't come together with any of the Centrapalus. Okay, so that was also nice. And then there are so many algorithms available to produce or to develop the phylogenetic tree using DNA or protein sequence data. And then it's often nice to use or compare these algorithms to see the outcome. And so we also use another program called Astral, so which uses coalescence algorithm to produce the phylogenetic tree. And then so this is the tree. And it also told us the very same story that the other analysis or XML analysis gave us. So here also it had a distinct Centrovirus excisions, a group for Centrovirus excisions, and then the earlier Centrovirus looking excisions, those were transferred to the new Vernolic group. They also clustered together as in the earlier analysis. So, however, close look at the tree. So there were a couple of unexpected outcomes. For example, these two nidobiences, so excisions. So we wanted them to, we expected them rather to come together, as in the case of this Australis. Here you can see that these two Australis excisions, they are together because they are most closely related to one another compared to the others. So similarly, we also wanted this here. Also, we wanted these Afromontana excisions to cluster together or to form a clade. And then, but those were not the cases. So we thought that, okay, so dense sampling might help us know what is going on here. So that's why we send more Centrovirus excisions from USDA for sequencing, and then we kind of combine them into a tree to produce this big tree. So with many Centrapalus excisions. So to focus more on the Centrovirus excisions, so we zoom in this, and then here it was interesting as per our expectations. So all these Australis excisions, this Lusotoensis excisions, or these excisions, they cluster together. They form their own cluster based on their DNA sequences, and together they formed. So together they form a group resembling, representing the subspecies pauciflorus as in our earlier earlier result. So similarly, the other excisions belonging to the belonging to these three subspecies, they cluster together, but it didn't serve our purpose because earlier we saw the nidobienses or the Afromontana clade not coming together, so not forming their own distinct group as they did here in Australis or Lusotoensis. So you can see that there's Afromontana from here, over here, and then similarly nidobienses also appeared at 2 or more places. So when such things happen, then the first thing we do is check our data. So we checked our data for any missing data in these excisions, but everything was fine in there, and then didn't have much missing data. So then we checked on the sources of DNA for these excisions. The green ones here, they are the ones that we grew in our greenhouse, in the University of Memphis greenhouse. The blue ones are the ones that that we collected the leaf tissues from the Smithsonian institutions and then museum, and then the black ones are the ones that we extracted DNA from a single seed. So if we take a look at here, even though these two Afromontana, the tissues collected from the Smithsonian, well identified, the plants look similar, but still like they come at different places. So which tells us that even though based on morphology, they are three distinct groups or three distinct subspecies, but that is not supported by the DNA data in our study. All right. So the next thing that we did was we produced the Microsatellite Markers, a genomic tool for the specific to Centrovirus species. So for that, we use the DNA sequence of the Centrovirus and then use the program Phobos, so which kind of identifies these short sequence repeat regions in the DNA, and it's an algorithm. And then we use another program called Primer 3, so which helps us to design the primers. So using those primers in the PCR reactions, we can amplify these regions and then compare them across the individuals in a population or across individuals in different populations. And based on that, we can perform the we can assess the genetic diversity and population structure in those populations. All right, so okay, so altogether we identified 30 microsatellite loci, or those short sequence repeat regions, and then developed the primers for them. And then 26 of them successfully amplified in all Centrovirus taxa. So here you can see that. So here there are different Centrovirus excisions in. So and then the microsatellite loci kind of amplified or in all the accessions. But here, so you can see that some of them were successful, they amplified nicely, but still others didn't do good. So that means we designed 30 microsatellite loci, 26 amplified successfully. And then the next thing was to check the variability in them, and only 10 of them had the variability. So that means if they didn't have any variations among the individuals in a population or among different populations, so they are not that useful um, for uh, for population genetic studies. So that means we had 10 altogether. We had 10 microsatellite markers that we could use for the study for Centrapalus population study. Okay, so using those 10 microsatellite markers, so we performed the population genetic study for the Centrovirus accessions. We took uh, 6 populations belonging to different accessions of Centrapalus, and these are the accessions that we had already used in the phylogenetic tree, and the seeds were collected from the Eastern African countries, originally collected from the Eastern African countries. All right. So and then so for each accession, we grew them using the seeds. We grew them in our greenhouse, and at least like 15 individuals per accessions were grown for the population genetic studies. All right. So based on the result of the population genetic studies, so the AMOVA or analysis of molecular variants, it shows 56% variation among those populations. So, which means that the higher this number, so it means that the populations are more distinct. They are not related, and then the DNA, their DNA is also different. So whereas if the number this among population variation percentage is low, so which means that there is not much distinction among those populations, and more or less they have the same DNA. So and then we have this 56%, which is kind of all right. So and then so we also performed the principal coordinate analysis. So where we could see that these small dots represent single individuals, and those there were 2 distinct clusters, which corroborated the results that we saw earlier in the phylogenetic tree. So, for example, these 3 accessions so they cluster together here, and then these 3 accessions from the next group, they cluster together here. All right, so we also perform the structure analysis. So this is the structure plot. These black vertical bars, they delineate the populations here. And then, each vertical bar in these populations, they represent the individual, and then their color kind of gives us the proportion of the proportion value that this individual belongs to one of the distinct genetic cluster. For example, like this different looking individual, so it belongs to this populations and that population, but this is odd here. So this result is showing that there are in our 6 studied populations, so there are 3 distinct genetic clusters. So earlier the tree phylogenetic tree, it showed us 2 clusters, but this population so it further divided one of the clusters into 2, making altogether there are 3 tight genetic clusters, as we can see here. So these 2 genetic clusters, those were identified by the tree phylogenetic tree, whereas these 3 genetic clusters were identified by the structure analysis. All right, so this table gives us the genetic variability. So for the 6 studied populations. So here, this parameter, which is called unbiased heterozygosity, so which tells that the higher the value means that there is more genetic variability in that accession, and the lower the value, obviously, is like there is low genetic variability. So it's advantageous to have the higher genetic variability. So so based on the results, we can see that this Centrovirus accession, so which is read as Centrovirus pauciflorus subspecies pauciflorus variety Peditiana. So that had the highest level of heterozygosity. So that means that there was more genetic variability present in that excrescence. And in fact, this is the excrescence that USDA earlier used to develop a day-neutral Centrapalus excrescence. So that could grow nicely in USA and then in Canada. So obviously, so high genetic variability might have been advantageous there. All right. So in summary, so we produce a highly resolved phylogenetic tree for Centrapalus species, showing our Centrapalus excrescence, showing relationships among different groups and subgroups. And then we also developed novel genomic tool for Centrapalus species for population genetic studies, and which will be available, which will be published, and then will be available for other researchers studying Centrapalus. And then also we assess the genetic diversity and population structure of some of the populations of Centrapalus pauciflorus excrescens. All right, so with that, I'd like to thank the Agriculture and Food Technologies Research Cluster for providing us with the fund for this studies. So similarly, I'd like to thank Department of Biology Sciences Center for Biodiversity Research for providing travel funds and then the research funds. So I would also like to thank High Performance Computing. So without their for giving us the opportunity or for helping us to run big volumes of data. So without their support, so running big volumes of data would not have been possible. So okay, also I'd like to thank this Smithsonian Institution and then USDA for providing us with the samples, MRC, UT for providing us the facility for fragment analysis. And then I'd also like to thank the members of my lab, Erica, Paige, Mike, and Jorge for helping me in the lab. And then I would like to thank Eric and Christian from the SPC, so for helping us for troubleshooting our SPC runs. So thank you with that, and I'd like to thank you all, and then would like to take questions.
Thank you, Ram. That was a very informative presentation. If any of our attendees have any questions, please feel free to list them out in the Q&A box at the bottom of your screen or in the chat button, and we'll be able to answer them. I say we, I mean Dr. Ram or Dr. Mandel will be able to answer them. So feel free to ask them away. Okay, looks like we have one question. It says, what are your thoughts on finding such high FST values in your population genetic analysis? And we'll just answer that. Thank you, Erica.
Yeah, so sorry, just a second. It's not working. Oh, okay, so sorry, I kind of missed that question. So could you tell me one more time, please?
Sure, give me 1 second. Let me
Yeah.
find it. It says, what are your thoughts on finding such high FST values in your population genetic analysis?
Okay, so that could be because so when you have low FST values, so there will be plenty of gene flow among those excrescens. So these excrescens are collected from different places. So so that could be one of the reasons, or like so I believe, and then maybe so there are so having high FST values kind of suggests that there is not much gene flow among those excrescens. So one of the reasons could be, yeah, so they are collected from different places.
Great, thank you so much for the answer. We'll give another minute or two if there are any more questions or observations anyone would like to give. Please feel free. Okay, I don't think we have any follow-up questions, except for the one that was really a great question. It looks like you must have answered everyone's questions throughout the entirety of your presentation. So I always take that as a good thing when there's very few questions. On that note, thank you everyone for joining. We do have more What's Next sessions coming up here in the month of May, but I'd like to give a special thank you to Dr. Ram, Dr. Mandel, and the CBio Center for the opportunity for Ram to present his ongoing research. If you have any questions, please let us know. Please email myself or anyone at the CBio for any follow-up questions. Thanks all. Thank you, Ram. Thank you, Dr. Mandel.
Thank you.
Thank you. Bye-bye.
Bye.